Hey there, welcome to the Basics lecture on photosynthesis. In our previous video, you learned how the high energy electrons in glucose are used to make ATP and then placed back on oxygen to make water. Today we'll take the low energy electrons on water and excite them back to high energy in the chloroplast and put them back on carbon to remake glucose. This equation should look familiar. It's the reverse of the one we saw in cellular respiration. Now we're reducing the carbon molecule and oxidizing water. The chloroplast is an organelle found in photosynthetic organisms like plants and algae. It, like the mitochondria and the nucleus, has an outer and an inner membrane. The space between is called the intermembrane space, and the central fluid is called the stroma. Membrane sacs, called thylakoids, are arranged in stacks called granum, and inside each thylakoid is the thylakoid space. There are three main inputs needed for photosynthesis: light, water, and carbon dioxide. And there are two main reactions: light reactions and the Calvin cycle. Let's take a look at each one. The light reactions occur in the membrane of the thylakoid, as shown here. Let me go over these main steps visually first and just watch, and then you can copy my text down on the slide that's on the next slide. It contains all the text. Okay? So the first thing that happens is light energy strikes the photosystem membrane protein here. And this causes low energy electrons from water, shown as the yellow balls, to be excited to a high energy state inside the photosystem. This high energy electron is then transported to a different protein, and this process allows the movement of hydrogen ions into the thylakoid space. The light energy then, the electrons go to the next photosystem. Light energy excites the electrons again to a high energy state, and this time the electrons are not used to pump hydrogen, but are instead placed on the electron shuttle named NADP+, and used to create NADPH. So high energy electrons are now stored on this shuttle. Our hydrogen ions, which have been pumped into the thylakoid space, are now allowed to flow through an ATP synthase, which turns ADP and phosphate into ATP, and this ATP, along with our NADPH, then progresses to the second part of photosynthesis. Here are the words for the things I just said for you to copy down. You could also label the diagram. Now let's look at the steps in a bit more detail. The membrane proteins that capture light energy are called photosystems. They have a central pigment molecule that are strong oxidizing agents and able to pull the electron off of water. Then the chlorophyll pigments around them absorb the light energy and use that energy to excite the electrons to a higher energy level. Again, here's the photosystem taking electrons, using the light to raise their energy. First system is used to pump hydrogens. Light energy again raises their energy, and they're then placed on an electron shuttle. This should sound rather familiar, by the way, after looking at cellular respiration. The second part of the light reaction is using those hydrogen ions to run through the ATP synthase in the process of chemiosmosis, again, just like we saw in cellular respiration. Both the NADPH and the ATP are set to the Calvin cycle, and you should see now that the work of light is to excite electrons and use that energy to make ATP and fill these electron carriers. Notice in none of this process has carbon or glucose had any appearance. You should be able to compare chemiosmosis that occurs in mitochondria with that occurs in chloroplasts. Use chapter 10 in your book to label this diagram, making sure you know which compartments in both molecules have the high concentration of hydrogen ions. The Calvin cycle itself is pretty straightforward. Essentially, we're building sugars. The main steps are to take carbon dioxide, to place them onto a carbon acceptor, to add ATP and NADPH to make them sugar, and then to rebuild the carbon acceptor after you've gotten rid of the sugar so that the process can go around again. So let's look at the steps in a little bit more detail now. There are 3 phases to the Calvin cycle. And in the first phase, called fixation, carbon dioxides enter the system and attach to this carbon acceptor molecule. That's the fixation part. During the reduction phase, which occurs here, we use the energy in ATP and the electrons stored in NADPH to add electrons that are high energy to these carbon molecules. This creates a high energy glucose and is reducing the molecules, which is why it's called the reduction phase. The sugar molecule is then released in order to become fully glucose and other organic compounds. We're left with this leftover carbon molecule that is regenerated using some more ATP to return to the original carbon acceptor state. So 3 phases. We fix the carbon onto the acceptor, we reduce the carbon, and then we regenerate the carbon. We started with this figure, and now we'll end our unit with it. Do you see that there was a cycle of oxidation and reduction from following the electrons around the system? You see that sunlight is used to energize the electrons, which are stored on glucose, and that mitochondria are able to break down those molecules, take the electrons back, and use the energies captured with them to make ATP, which can be used in cellular processes. And I want to end also by putting chloroplasts back in the plant cell. Plants are able to harness sunlight to make glucose in chloroplasts, but remember that plants also have mitochondria. They utilize glucose as a stored fuel just like animal cells and can run cellular respiration just as well as any animal can. Plants don't do photosynthesis instead of respiration. Plants do photosynthesis in addition to respiration. This completes our basics video. Write out the answers to these questions to study for the quiz.